Have a short break. She comes from County Meath and she went to NUI Galway to study microbiology and continued her interest in microbiology and in the marine. She also happened to spend part of her career as a climbing instructor in the United States. So she does a lot more than just science. But with her talk on barnacles, bacteria and beyond from NUI Galway, please welcome Joanne Duffy.
My name is Joanne, and I'm a kleptomaniac. I steal genes, but not like genes, like your genes. A gene is a biological instruction. It's like a computer code that's written in DNA that tells a cell to manufacture a specific protein. Now, some plants and animals produce really interesting proteins, things like antibiotics and compounds that can fight against cancer, and even just things that can make your food taste really, really good. My talk to you today comes in 3 parts: barnacles, bacteria, and beyond. Barnacles. Barnacles are marine organisms that are sessile, which means they don't move. They don't move because they produce a really sticky glue that allows them to stick to just about any type of surface you can imagine, man-made and natural. Now, this glue could be really useful in everyday life and in medicine. So you'd think that if we wanted to use the glue, all we have to do is grow up lots of barnacles, harvest the glue at the end, and then we have it. But that's a disaster waiting to happen, because barnacles can cause huge environmental damage, because they really do stick to everything. They stick to buoys, they stick to boats, they stick to submarines, they stick to sensors. It's just a sticky situation. We can't effectively grow barnacles in the lab either, which means, like them, we're stuck between a rock and a hard place. But remember, a gene is a biological instruction. So what if we could take the gene for the glue and leave the barnacles behind? What if all we needed was the genetic instructions to make the glue? Well, when you're studying, you don't write down every single word from a book. You just write down the important parts. And a cell is just the same. It doesn't use all of its DNA. It only translates some of it into protein. And first, it turns it into a molecule called RNA. So I take glue from barnacles and I look for the RNA. I look and try and find, what did the cell bother writing down? And then I can literally make the gene for barnacle glue in the lab. I want everybody to close their eyes. Try and imagine all 7 billion people on planet Earth, lined up whatever way you like. It's basically impossible. Humans are hardwired not to be able to think of numbers like that. You can open your eyes. There are more bacteria in your mouth right now than there are people on Earth. Now, that's not meant to gross you out. My point is they're really small. They're so small that we can fit billions and billions and billions of them in just a tiny flask. So remember that gene I made earlier on? If I gave that gene to a bacterial cell, do you think it cares where the DNA came from? Or will it just follow the biological instructions? This guy doesn't care. He's going to follow whatever instructions you give him. He's having a party. So if I hand over that DNA from a barnacle to a bacterial cell, it'll follow the instructions regardless. Now, some proteins are too complex for a bacterial cell because they're too big or they've got all sorts of complicated sugars attached to them. But in general, DNA is just like computer code. Whatever works on one computer works on another computer. Whatever works in a barnacle will work in a bacteria. So by genetically engineering bacteria to produce the glue from barnacles, it's like we're able to hack into the greatest computer system of all time, evolution. Evolution has provided us with basically a limitless supply of genes that manufacture just about any product that you can put your imagination to, some of which can change the world. The bacteria that I use double every half an hour, which means that if I start out with 1 million cells, I end up in 24 hours with lots. I have lots of cells. Beyond. Well, what's the point of making glue? Well, barnacles live underwater. So we're hoping that this glue can be used in the presence of all kinds of fluids, like blood, or fluids that might need to be used during a surgical procedure. That means that we could revolutionize the way surgery takes place, and we could try and glue your skin back together. We could glue wounds closed. We could cement a medical device in place in your body. This technology might sound a little bit like science fiction, but it's actually well established, and it's used to make products you're familiar with. Chinese food tastes really good, yeah? The reason it usually tastes so good is because it contains an addictive salt called MSG, or monosodium glutamate. Now, monosodium glutamate is made in the sugarcane plant, but back to our bacteria earlier on, sugarcane plants take up quite a bit more room than bacterial cells. So scientists steal the gene from sugarcane plants and put it into yeast and bacterial species and force it to make the salt for them. And it's exported, and it's a multi-billion-dollar industry on a global scale. Anybody here suffer with diabetes? Diabetes type 1 used to be an early death sentence. It was really, really dangerous. So when you have diabetes, your pancreas doesn't correctly produce a hormone called insulin, which lowers your blood sugar. It's because your immune system is busy attacking your pancreas, which is just rude, to be honest. So scientists used to have to take insulin from a pig and inject it into people. Now, you should never put anything from a pig into your body unless it's bacon. On reflection, I should have called this talk Barnacles, Bacteria, and Bacon. Scientists took the human gene for insulin and put it into a yeast species, and because it's been given the human instruction for insulin, it makes human insulin. And that now lengthens the lifespan of people with diabetes tenfold. So, I'm a genetic kleptomaniac. I steal genes from organisms that I've sometimes never even seen. And for now, I'm sticking with glue. Gurmila Mahagi Sakdeisha Club.
Okay, thank you very much, Joanne. So over now to our panel for some questions.
Thank you very much, Joanne. Very entertaining. Just have you experimented with applications for glue that might emerge from barnacles?
Yeah, so the really interesting property about the glue is obviously that it works underwater, because traditionally an adhesive, you know, if you leave a bottle of glue open, it'll dry up. It actually is dissolved in a solvent, and when the solvent evaporates, that's what allows the glue to harden. So what's really interesting about, I love barnacles. What's really interesting about barnacles is that it's the opposite. It actually seems to require the water for the glue to cure. And it also takes in ions from the water, so it depends on where the species have evolved, what kind of interactions there are. So there's one specific one that has calcium-specific activity, so it needs calcium in the water, which would be very interesting in terms of gluing bones back together. So yeah, it's in its early stages now, but there's a whole host of applications down the road, yeah.
I'm still trying to get over the 7 billion bacteria in my mouth moment. So gross. But what about when you're trying to dissolve? Can you dissolve the glue?
That's a very good question.
You get it wrong and you suture the wrong vessel or whatever.
That's the other interesting property about it in terms of using it in surgery, is that you can degrade it, but it's bloody difficult. It's really, really hard. So the US Marines have a patent on a particular compound that they put in the paint that they put on their boats that deters barnacles from sticking to them, but even that doesn't fully work. They're really notoriously difficult to get rid of, so they stick to all types of surfaces. Some species have a specific type of surface that they'll stick to. Sadly, some of them have actually evolved to only stick to plastic because of the amount of plastic that we put into the oceans. But there are ways to degrade it. So that's the other. One side is looking at building it up and using it in biotechnology, and the other golden ticket would be finding an easy and non-hazardous way to degrade it in the oceans that wouldn't harm other marine life.
Thank you. It would appear that the main thrust of your research is around the medical possibilities around this. Have you thought, or is there any incidental industrial kind of applications you might get through this? Yeah,
so the reason that I'm so interested in the medical side is just purely based on my research group. So it's a biomedical focused research group. But yeah, there are lots of industrial applications. So for example, underwater computers or underwater cameras and phones might require adhesives to stick chips in place, and that's where this kind of adhesive would come in extremely useful. So yeah, there's a whole other area of applications. You're dead right, but we focus on the medical stuff. And you know, 4 years seems like a long time for a PhD, but the race is already on. So yeah.
Okay, so for her talk on barnacles, don't be shellfish, and please give Joanne a big round of applause. Okay, we're now going to have a 5-minute break. I mean 5 minutes, so if somebody does need to go to the bathroom or you have to do anything urgent, you can. Do be back in 5 minutes. We have 2 more excellent presentations, and then you have to make the decision.